Hello, everybody, and welcome back to Creation Myths. Before I get into tonight's topic, I'm just going to make sure the sound is okay on my end. And once I confirm that we sound okay on YouTube, then I will get going. It seems okay to me. So let's do this. Hi, everybody. Welcome to another installment of Bad Creationist Genetics. We've hitting the series a lot lately. So today I have a super fun one for y'all, and we're just going to go through it together. This is a short video. It's only about 15 minutes long from the Institute for Creation Research, ICR. And this is an episode of their podcast. They put this, all these interviews and stuff up on YouTube. So that's where I consume them. And this one is, this particular episode features Dr. Jeff Tompkins, and the topic is junk DNA. And the argument that is going to be put forth by Dr. Tompkins here is that junk DNA is not real. And that essentially 100% of the human genome is functional. So that's the argument we're going to get. And we are going to see just how bad this is, because it is quite bad. So I'm just going to let kind of the intro bit play for just a minute here to introduce the topic and everything, and then I will periodically pause and point out some of the major problems here. And then once we finish the video, there's kind of a funny addendum to this that I'll also show you. So we'll go through the video. Like I said, it's about 15 minutes long. We're not going to watch every minute of it. I'm going to jump around to kind of relevant parts, and there's parts that I'll skip because they're just not relevant. And then after the video, we'll do a quick kind of wrap up thing that'll show you kind of how confident ICR is in what they have going here. So with that said, let's get it started, and I'll periodically pause and jump in when I want to correct something. So here we go. Oh, when I start the video, for those of you in the future, hi, you can ignore what I'm about to say next. For those of you watching this live, hi, welcome, thrilled to see you here. Let me know if the audio in this video right here is good. The level is good for me, but if it's too loud or too quiet for you, let me know in the chat so I can adjust it as necessary. With all that out of the way, let's get this started.
Hello everyone, welcome to the Creation Podcast, a show where we discuss the science that confirms Scripture. I'm your host, Lauren, and I'm here today with ICR's geneticist and Director of Research, Dr. Jeff Tompkins. It's so good to have you here today, Dr. Tompkins.
Wanted to jump in a little bit about Tompkins here. A little quiet, I'll turn that up. There you go. A little bit about Tompkins here. He is well known for his extremely bad analysis of the human chromosome 2 fusion, and also his extremely bad human chimp genome similarity comparison. It's just terrible. Other people have talked about it elsewhere. I'm not going to go through the details here, but just Google it, look it up on YouTube. It is amusingly terrible.
Tompkins.
It's great to be here.
So today I wanted to talk a little bit about junk DNA, or rather the DNA that many people try to pass off as junk. From my pretty basic knowledge of the topic, only a small portion of any given creature's genome is responsible for coding for proteins. That leaves a lot of DNA left over, and many people try to say that that is completely useless or junk.
Right away, that is wrong. That is not true. In the human genome, a little bit under 2% of the DNA is exons. That's the regions that code for amino acids to make proteins. So what creationists often either straight up claim or just indirectly imply is that biologists thought at some point that the other 98% of the genome wasn't functional, was junk DNA, 2% versus 98%. That's not true. That has never once been the case. We've known about regions that are non-coding, that are also very important functionally, since before the term junk DNA existed. Two simple examples are tRNA genes. We've known about tRNA genes since we figured out how translation works. That was before the term junk DNA was coined. The term junk DNA wasn't coined until the 60s or 70s. It was related to neutral theory. But we've known about tRNA genes since well before that. Those do not code for proteins, but they're unquestionably functional. We've also known about things like promoters and operators, regulatory regions for gene expression. Those don't code for proteins. They're unquestionably functional. Those were first kind of figured out in the early 60s in a classic series of experiments. So right off the bat, we have this problem set up where we know that 2% of the genome, a little less, codes for proteins. And then, but what about that other 98%? People think it's junk, right? No, that has literally never been the case.
So where did that idea come from?
Well, we have to go back in history to really the late 1960s to see where that idea came from. So evolutionists began proposing a model called the neutral model of evolution. So this idea basically says that much of the genome is just neutral. It's free to evolve and pop out new genes and new sequences.
For the record, that's true. We've directly documented that is the case. And the way you can document this is show that there are regions of the genome that are not subject to what's called purifying selection. Purifying selection is natural selection that removes new variants. Purifying selection keeps a sequence the same. If a sequence is not subject to purifying selection, one of two things can be the case. It could be under what's called adaptive selection, so you're getting selection for new variants in that sequence, or it can be unconstrained. It could be evolving neutrally, right? Those are your two options. And we've directly documented that there are portions, large portions, the majority of the human genome that is not subject to purifying selection nor adaptive selection. It is evolving neutrally. So what Dr. Tompkins just said here, or what he kind of implies, that there's no neutral regions, that's wrong.
Would, provide something for the mystical agent of natural selection to act upon.
I know I promise you I will have longer segments that'll let it go as we go, but I just need to comment on this one line. This mystical agent of natural selection. Remember, this video is coming from ICR. ICR's kind of pet thing is the environmental tracking, the consistent environmental tracking, and CET. And it's this alternative mechanism of adaptation to natural selection. And it's totally wrong. I've done a video on it. I'll link that below in the description. We're not going to go into it here, but it's a thing. CET. I think I got the acronym wrong. It's something environmental tracking. It's wrong. It's bad.
And so this was before the days of DNA sequencing.
Continuous environmental tracking. Thank you. That's why I have an audience. Thanks. That's why you're here. Appreciate all of you. Continuous environmental tracking. Thank you.
Sequencing even. So that's where it really got going, was back then. But then as scientists began to develop DNA sequencing, which was very crude in the beginning, in the 1970s and the 1980s, they began discovering that genes, especially in what we would call eukaryotes or creatures that have a nucleus and have individual chromosomes, they began discovering that genes were in pieces, as they originally said. So you would have a gene, but only certain parts of that would code for protein, and the intervening parts did not. And they originally thought, oh, this is junk, and it fit well with the paradigm of neutral evolution.
I want to just pause right there because this is a really nice illustration of the way that credentialed creation scientists can be dishonest. So what he just did is he summarized a finding in very specific terms, very precise terminology in such a way that he wasn't inaccurate, right? What he just described was introns and exons. He didn't use those words. When you have a gene in the human genome, right, in eukaryotic genomes in general, you've got a place where you start making mRNA from it. Okay, you got a transcription start site, you've got a long gene, and they've got a transcription stop site. Now, the RNA you make is not a mature mRNA. It's got parts called exons that are protein coding, and it's got bigger parts in between called introns that are not protein coding. And what he just did is he described the introns without using the words. And he did it in such a way that makes it seem like the argument from biologists is that anything that's not protein coding is junk. But that's not the point of the finding he described. What the actual consensus was at the time was that these introns are junk. We now know that there are sometimes things like regulatory sequences within introns. You could have an enhancer for one gene in the intron of a different gene. That's a thing we know exists now, but we didn't know that when we first discovered introns. So what he's doing is he's taking an old finding on this, just the existence of introns and exons, and he's presenting it as though it means biologists at the time, and we're talking about the 60s and 70s here, right? That's the kind of time frame he gave, thought that all non-protein coding DNA was junk, and that's fundamentally dishonest. But he's doing it, again, in a very precise way where the specific words he says are true, but the meaning is not, right? And it's if you watch other credentialed creationists doing this, other people with PhDs, Jeanson will do this, Behe will do this. It's a very common approach they take to explaining findings, where they will explain it in very narrow, very particular terms in a way that is accurate in the small sense, but it gives the incorrect picture in a broader sense. It's just very devious, and everyone should be on the lookout for that.
And then as DNA sequencing began to progress, they began discovering even more parts of the genome that did not code for protein. So when we get to the Human Genome Project, which really got going in the 1980s and is actually still going on today, they began discovering that less than 2% of the entire human genome actually coded for protein.
Wow.
And so that began to fuel the idea of this neutral model of evolution that was begun in the 1960s, and it seemed to validate it.
Mm-hmm.
So that last line is actually accurate. That finding that most of the genome was not just not protein coding, but not constrained at the sequence level, that did validate the neutral model of evolution, right, at the molecular level. That was a prediction, actually, of neutral theory in the 60s, 70s, into the 80s, predicted that when we do sequence the genome, we're going to find a lot of regions that are not constrained at the sequence level. That was a specific prediction, and that turned out to be accurate. Now, some of the other stuff here that I just want to comment on very briefly is that this does not really fuel the neutral model. By the time you got to the Human Genome Project and sequencing the human genome, we're talking late 90s, early 2000s, the neutral model was well established. We had lots of evidence that the neutral model was a valid approach to looking at this, including experimental evidence, where you could look and just see, like, is a sequence under selection or is it evolving by genetic drift, neutral processes? And the answer is large parts of the genome are evolving neutrally when you look experimentally at that. So we didn't need the Human Genome Project to fuel or validate neutral theory. Neutral theory by the turn of the century was well established. So it's not that it was kind of on the ropes until we saw this 98%, 2% thing in the human genome. It was well established before then.
That would seem very validating at first glance, but is all of that DNA, the 98%, is all of that useless? Is it junk?
Okay. He actually goes off topic here, so we're gonna jump ahead. But I do want to just point out that question doesn't even make sense, at any point, because when the term junk DNA was coined, which I believe, again, was in the 1970s. I forget the specific year, but I think it was in the 1970s. When that term was coined, we already knew about a bunch of parts of the genome that were not protein coding that had specific important functions. So at no point Did anybody say the breakdown functional, non-functional was 2%, 98%? We already knew that was wrong when the term junk DNA was invented, decades before we sequenced the human genome. I just want to point that out. It's just, this is, again, something typical coming from a creationist organization, that you get these questions that are based on like ignoring decades of biological research. So now we're going to jump ahead to 350 here. Here we go. Jumping ahead because we don't have all night. We got to hit the high notes here.
They actually found out that almost the entire human genome was being transcribed or turned into RNA end to end. In fact, one paper called it a RNA machine. The human genome was an RNA machine. And so it looked like the entire human genome was functional. There was RNA being transcribed from one end of it to the other.
Okay. This is entirely wrong. The human genome is not entirely transcribed almost from end to end, his words, and then he left out the almost on the second time there and just said from one end to the other, it's transcribed. It's not. A large fraction of the genome is transcribed. According to ENCODE, it's somewhere between 70 and 80%. The ENCODE project is like the what fraction of the human genome is functional project that grew out of the Human Genome Project. And according to their findings, somewhere between 70 and 80% of the genome is transcribed to RNA. Their 2012 paper put it somewhere in the 70s, I think, and then it's been revised a couple times. It was somewhere in the 70% range. Now, I'll note that a lot of that is at extremely low levels. You're talking countable numbers of transcripts per cell, right? So the takeaway here is twofold. One, the facts of the statement are just wrong, right? The genome is not transcribed end to end. In fact, a lot of the genome is persistently in a form called heterochromatin, which is very densely packaged DNA with proteins. And there's some parts of the genome that are heterochromatin in every cell in your body. And they're just densely packed. It's proteins with DNA wound around them. And then it's this kind of ball of DNA and protein called a nucleosome with a linker region, and then another nucleosome. And it's this kind of chain of DNA wrapped around proteins. And then that fiber is supercoiled. It's densely packed itself. The effect of that dense packaging is that the proteins required to transcribe the DNA into RNA can't actually access the DNA. So it's just not the case that all of the genome from end to end, or almost all the genome from end to end, is transcribed to RNA. That's the first problem. The second problem here is that his argument is that, well, if it's transcribed, that means it's functional. That's not true. You know who says it's not the case that transcription equals function? You know who says that? ENCODE says that. In their 2014 paper, that creationists pretend doesn't exist, ENCODE was much more circumspect about the implications of their findings. One thing they talk about in that 2014 paper, which is linked down below, they talk about this in great detail. And I really appreciate the level of nuance in the discussion in this paper. They talk about how it's hard to parse when RNA transcription is actually going to be a sign of function, because some transcripts are at extremely low levels, to the point where you would only have one or even a few copies of that transcript in the cell at any given time. And at that level, it's just physiologically not going to matter. It's not going to have any effect. And if it's not having any effect, how could it be functional? They also talk about conservation of these sequences that are transcribed. In other words, are they under purifying selection? Is there natural selection acting upon those sequences to keep them the same? If there is, that's a pretty strong signal that they're functional. But for most of these sequences, for most of these transcribed sequences, they're derived from something else we're going to talk about a little bit later on here. They're actually not constrained, and we can demonstrate that they can mutate freely without a fitness cost, which is strong evidence that they're not functional, or at the very least not functional at the sequence level. Okay, so there's two layers of wrong here. There's the direct layer, if he's just misstating the data that we have, and then there's the implication that transcription equals function, and that is also incorrect. Let's continue. Let me make sure. What are we up to? We're up to 415. Okay, so now we're going to jump ahead because there's a part in here where he goes through like what RNA is and what transcription is, and it doesn't matter. So we're going to jump ahead about 3 minutes to get back to the everything is functional argument. Okay, that'll be fine.
They do not code for protein, they code for functional RNA,
It's the tail end of the last thing.
or they might have regulatory regions around the
Here we
genome that don't code for protein as well. So these would be like, say, like a light switch. So when you walk in a room, you flip the light switch and it turns on the lights. Well, there's a lot of regions in the genome that are basically switches and control regions. In fact, there's millions of them in the human genome that turn off and on things, that tell certain parts of the genome what to do, that turn on genes, that tell the genes how much product or RNA to make, how long to make it, how fast to make it. And so it's very complex what's going on in the genome with all of these regions that do not code for protein, that were once thought to be junk.
And that last line is where he stuck his foot firmly in his mouth. Because what Dr. Tompkins just described there was things like promoters and operators and enhancers and silencers, regulatory regions in genomes that are very important for the regulation of gene expression and other functions, are not transcribed into RNA, are not translated into protein, but are unquestionably very important functionally for the proper workings of the genome. Now, we just said at the end that we thought all that stuff was junk at one point, and that is a bunch of nonsense, because those regulatory regions, those were first kind of, I don't want to use the word discovered, but they were kind of figured out for the first time in a series of experiments that were published in 1961. The term junk DNA, again, didn't exist until the 1970s. So we knew about these regulatory regions that he just described. We knew about these regulatory regions before the term junk DNA existed. So at no point in history were those regions thought to be junk DNA. He's just lying straight up. Like, it's not more complicated than that. I just want to take a second to say thank you to One More Once. Oh, there you go, teaching me stuff again. I appreciate your generosity. Thank you. I appreciate that. Alrighty, so let me check my timestamp here. Okay, so that was there. Great. We're going to keep going because the next thing we're going to talk about, you'll see, it's a good one.
Wow, that's amazing. That's amazing. Kind of changing gears just a tiny bit. What about pseudogenes? Can you tell us a little bit about that?
Yeah, pseudogenes were genes that had similarities to protein coding genes. And scientists at first thought that they were junk because they didn't code for protein. Now we know that almost every pseudogene that's been studied is actually a functional RNA coding gene.
So that's wrong. That's wrong on two levels. One, many pseudogenes don't actually make an RNA. What pseudogenes are, background for everybody, a pseudogene is when you have a gene that mutates in such a way that it can no longer function as a protein coding gene. Maybe it mutates and it has a stop codon, like really early in the sequence. So you start translating it and then it just stops and you get this little end of the protein and then there's nothing. Or maybe the promoter breaks or the start codon mutates, so there's no start codon. But the sequence is clearly recognizable as homologous with a functional gene in your genome. Oftentimes, pseudogenes are the product of gene duplication events. So you have a gene, it has a function, it's under purifying selection. It duplicates. Now, one of those copies needs to work. You need that gene, but the other copy, if it mutates, there's no cost to it because you already have, a backup, right? So then you get what's called a pseudogene, a non-functional version of a formerly protein coding gene. That's what a pseudogene is. Okay, so he's saying almost all pseudogenes code for functional RNA. Now, that's not true, one, because many pseudogenes don't make RNA at all. Very famously, the gulo pseudogene in humans, this is the vitamin C. This is why we can get scurvy, because our vitamin C biosynthesis pathway is broken because of the pseudogene. That gene doesn't even have a promoter. No promoter, no transcription. Like just straight up, you're not making RNA from that. So it can't work. But the second thing is, as I said before, being transcribed to RNA does not automatically mean you're functional. You need to back up that assertion on a, for pseudogenes, case-by-case basis. If you want to demonstrate that a specific pseudogene has a specific regulatory function, you need to document, one, that it's actually transcribed, two, that mRNA is present at a level to be biochemically relevant, and three, if it's having a regulatory function, it's going to be constrained. It has to be conserved at the sequence level. It's going to be under purifying selection. If it doesn't check those three boxes, that thing isn't functional. And you have to do that for each pseudogene. And he's claiming that, oh, we've checked and they're all functional. No, they're not. That's wrong. Just wrong. The pseudogene stuff, I'm going to let this keep going for a little bit more because we have more to hear here, and then I have more to say about pseudogenes.
In fact, many of these pseudogenes produce RNAs that actually regulate the protein coding gene that pseudogene is similar to. And so pseudogenes are turning out to be very important for human health. When there's mutations and problems in certain pseudogenes, it can lead to heart disease and diabetes and all sorts of different health problems in humans. So pseudogenes are now actually, most of them that we have studied are actually functional, what we call long non-coding RNA genes that produce functional RNAs that do really important stuff in your cell.
That's not true. That's not true at all. So in order, what he's describing is a mechanism, and again, he's not like providing enough details for someone who doesn't like really dig into this stuff to make sense of it. But the mechanism that he is proposing is that you have a gene, a functional protein encoding gene that is transcribed to mRNA, right? This other molecule. And then you have a pseudogene that's similar to it. And that pseudogene is also transcribed to RNA. And that very similar RNA somehow regulates the subsequent activities of that functional gene mRNA. And there's a couple hypotheses about how this could happen. And some of it probably is accurate, but not in every case of every pseudogene. But the mechanisms would be like this. One is you could have direct interaction between the two mRNAs, and that could potentially slow down the rate of translation. You could also have the pseudogene RNA acting as a decoy for things that might target the correct RNA for degradation. And that's going to increase the lifespan of that mRNA and therefore increase the amount of translation. Now, a couple things here. In order for that mechanism to work, those two sequences have to be extremely similar to each other, which means the pseudogene needs to be under purifying selection. That sequence is going to be constrained. And if it's not, then it's not doing this proposed activity. Well, we've checked this. We can look at pseudogenes in the human genome, which don't make up a lot of the human genome. We're looking at less than 1% here, but it's not zero. But we can look at these pseudogenes, like the gulo pseudogene involved in vitamin C synthesis, and we could see that it's accumulating mutations. It's not under purifying selection. You could do this for other pseudogenes as well, which means that this proposed function that Dr. Tompkins is describing is simply not the case. The other thing to consider here is that there's a very frequent conflation with activity and function in creationist circles when it comes to evaluating the functionality of the genome, and also a conflation of factor A affecting thing B and factor A regulating thing B. And those are two different things. So, and the difference is selection. If something is regulatory, it's going to be under selection to do that regulatory activity. But something can just have some spurious interaction and affect something without being selected to do it. Picture, for this analogy, think about it like this. You've got, your house. In the winter, it's cold, so you turn your heat on. Now, it turns out in your heating system, you have a vent that is blocked, right? Some of your ductwork is blocked. So the warm air that's supposed to be going into your house, it's actually not going into your house at the level it's supposed to. It's supposed to be going this much, but it's actually lower than that because one of your vents is blocked, right? Hypothetically. So say you want your house to be 21 degrees Celsius, right? Well, you maybe have to set your thermostat for 23 in order to achieve 21 because you've got a malfunction in your heating system here. That's similar to what's going on with these interactions between pseudogenes and their functional counterparts, is that there may be an interaction there. There may be something that is causing a modulate 21 is too hot. Yeah, 19, whatever you find comfortable in your house, and this is Celsius, people. If you're working in Fahrenheit, don't set your house to 19 degrees. That'll be very unpleasant. So if you could have these interactions and these effects that the cell has to account for. Maybe it interferes with the functional mRNA. So to compensate, you upregulate the transcription of that mRNA. Now, if you were to change that, if you were to have mutations that cause that interaction to be different, that may very well cause a problem. That could cause a disease. That doesn't mean it's functional. You could also have a mutation that causes a pseudogene to maybe be more active when it shouldn't be. Maybe that causes a problem. So the fact that these can be, I should say are, because very few of them actually are documented to be disease associated, but like being transcribed, being disease associated is not on its own good criteria for a good criterion for functionality. You need to match that with other things. So there's a bunch of reasons why what Dr. Tompkins just said here is incorrect. But I think the simplest one is just most pseudogenes are not constrained at the sequence level. They're not subject to purifying selection, which means the proposed regulatory functions aren't actually happening. And it's not more complicated than that. So let's continue. That was, where are we up to? We're up to about 9:23. Okay, so we're just going to let this play because we're going to do the next thing is transposable elements.
We're finding these elements that we thought were just kind of throwaway are actually completely crucial for various things that our bodies are doing.
Yeah, absolutely.
Wow, that's amazing. So can you talk a little bit about transposable elements and what that looks like?
Well, there's many different types of what's called transposable elements. At first, they were called jumping genes. And so transposable elements are in different categories, and it can get very complex in trying to explain all of the different categories. So I'll try and make this as simple as I can. Most transposable elements are actually not jumping around or doing anything like that.
So are people...
Okay, I just want to pause there because that was incredible. The question was, what are transposable elements? And the answer was, well, they used to be called jumping genes. There's a lot of different types of transposable elements, but it turns out a lot of them aren't actually jumping around in the genome anymore. And that is not an answer to the question. And I have a hypothesis why he didn't give an answer to the question. And the answer is because he can't accurately describe what transposable elements are without kind of going against the ICR company line, right? The company line, not just for ICR, but for most young Earth ministries, right? Most young Earth organizations is going to be that transposable elements are functional elements that were created in the genome. Now, Dr. Tompkins here, they introduced him as a geneticist. I forget what his PhD is in specifically, but he does genetic stuff. He knows what transposable elements are. They're the remnants of mobile genetic elements in our genome. They're essentially the remnants of genomic parasites, right? These elements that get into a genome of a cell, they copy themselves, they cut and paste and put themselves, or copy and paste. If they're retrotransposons, they copy and paste themselves all over the genome. And in our bodies, over 40% of the human genome is transposable elements. It's a huge fraction of the human genome. And they get there because there are these elements that just copy themselves and move around and copy themselves. Most of them are no longer mobile because, one, they've mutated, and two, we've inactivated them, either through our immune system that recognizes foreign DNA and mutates it so it can't be functional, and also through methylation, where we methylate our genome, and like I described earlier, it curls up very densely, and so it can't be expressed, so they can't move around. And And also they just mutate spontaneously, and they break and they get stuck and they can't mobilize anymore. Dr. Tompkins can't say any of that, because if he explains like what transposable elements are, that's he's going against what, like, I don't even know what the official ICR position is, but my guess is that it's the typical young Earth position, that these were created in place with specific functions. So he didn't answer the question of what they are. Now, I'm going to skip the next part because he talks about what they kind of are doing, without kind of touching on functional stuff that is important for this broad question. So we're going to go up. We're going to actually skip quite a bit here, which is good. So we're going to go up to 1230 because this is already taking longer than I anticipated. So weird how that happens, right? So let's go up to 1230 here. What are we going to talk about at 1230? Oh, this part's good. Yeah, you'll like this.
Very fearfully and wonderfully made.
No, exactly.
So if you're thinking along the lines of evolutionary theory, then over that long of a period of time, all of that non-coding DNA would be mutating, evolving.
Right.
Do we see that happening?
No, we really don't, because when we compare, say, different types of mammals and their genomes, a lot of these regulatory elements that are non-coding are very similar between these creatures. And so they're not showing any signs of evolution. It's just common code serving a common purpose, just like a computer programmer would use. And so I actually have had to do computer programming in my research. And when I create a new computer program.
Okay, so I want to pause here, and then I'll let this little computer programming piece play, and I'll talk about something there. So the first thing here is that the question was, is the non-coding part of the genome evolving? And he says no. And that is bananas. That is nuts. We can document polymorphisms in human DNA. Like in the human, we've sequenced a lot of human genomes now, and we can show that a lot of the human genome has polymorphisms. There are mutations that occur. That's evolution, right? Evolution is heritable changes over generations, right? You're changing the genome sequence over generations. That is unquestionably happening, especially in the non-coding parts of the genome, because those parts of the genome are not constrained. Again, we can go to Encode for this. In their 2014 paper, they have, I think it's figure 1 in that paper. It's the 2014 paper linked down below. I think it's figure 1. They have a Venn diagram showing different levels of like how you might ascribe function to different parts of the genome. And the part that is conserved across evolution is very small. Like for mammals, like 5% of the genome, for example. It's not a lot. That's like constrained across all these different groups. And the reason for that is that most of the genome isn't actually functional. And what he describes here is like, we actually see a lot of similarity between like humans and other primates and mammals. And it's like, yes. And if he was more specific about this, I talked about the degree of similarity you see among increasingly broad groups. You know what he's describing, everybody? He's describing nested hierarchies. This is one of the best pieces of evidence for evolution, and he's treating it as, oh, no evolution here, nothing to see. It's just mind-boggling that he could take something that is just straight up, like, here's a red flag, and I'm just going to straight up tell you it's green. Like, 100%, I'm just going to lie to you. And that's what's happening here. This is just astonishingly dishonest, that bit that I just played. So now he's talking about like how, oh, it's just shared pieces of a computer program. So let's let that run a little bit, and then I'll talk about that for a sec.
To do something, I don't create a whole bunch of new code most of the time. I borrow code from other programs.
Right, because it already exists.
Right, it already exists. And so I just put it together into a new system. Mm-hmm. Now, definitely creatures have their own unique DNA. So humans have DNA besides the common elements that do common things between, say, mammals. But humans also have their own unique repertoire of DNA sequences as well that are regulatory, protein coding, and the various types and categories of DNA that there is.
Also, I just want to point out, kind of related to the last thing I said, of this just rank dishonesty here, before I get to the actual computer program reusing functional parts thing, is that I think you can hear it in his voice that he knows he's lying. Like, you could just hear that it's crushing his soul because he knows better. This is a man with legitimate credentials. He knows that what he's saying is dishonest. And I think you can, not to like go, psychoanalyst on him, but I think you can hear that here. The other point I want to make with this this is essentially a common design argument, right? Oh, they're similar in these different groups because it's the designer reusing functional parts that are doing the same thing. We have a direct test of that, which is looking at non-constrained sequences and seeing the extent of nested hierarchies among non-constrained sequences. Are they limited to what creationists would call created kinds, or do they span across created kinds? I'm not going to go into the details here because I have a whole video on it, and I will link that below after I do this. But the when you test that hypothesis, again, the evolution and the creation predictions, the creationist prediction is just wrong. The nested hierarchy span across created kinds, and that's just a direct refutation of separate ancestry. So that's worth pointing out here. Okay, so now we're next time point is just right here, and we're almost done, everybody. We're just about there. Thank you all for hanging in on this. We've been going for 40 minutes, and we're almost done with this video.
Wow, that's amazing. Thank you for touching on that. So over the last couple decades, humanity has increased a lot in our knowledge on genomics, and also with advances in technology, it's made it a lot easier to study this subject. So what do you think, you've touched a little bit on this already, but what do you think just the top takeaways are? What are the most important things that we've discovered about genomics, especially in pertaining to junk DNA here in the last couple decades?
Well, I think one of the most important things we've discovered is the immense functionality of the genome from one end of it to the other. So there are not regions that are just sitting there neutrally evolving, neutrally mutating. It's just not happening.
I'm sorry, what? There are no neutral regions in the human genome? None? That's something. That is something else. I mean, that is just, no. There are unquestionably regions in the human genome that are neutrally accumulating mutations. And to say otherwise is, again, it's just dishonest. This is completely bonkers. Like, what are we doing here, Dr. Tompkins? You know better. You know that that is not true. There are neutral regions in the human genome. We can document, like, within protein coding genes, there are neutral regions. Within introns, there are neutral regions. In pseudogenes, in endogenous retroviruses, in transposons, tons and tons and tons of neutral regions that we can directly document are not subject to purifying selection. That's what neutral means. And we can show that that is the case for huge swaths of the human genome. It's just, I like, it's almost like I'm almost speechless hearing somebody, again, who is like, sure, like Joe Schmo creation is going to say stuff, but like Dr. Jeff Tompkins knows better, and he's saying it anyway. And that is, it's shocking, but not surprising, I think we'll say. I think that's a good way to put it. All right, we're almost done here. I promise. Let's keep going.
In fact, many scientists now are realizing this, and it's creating a huge problem for evolution because the entire genome now is appearing to be functional. And areas that were thought to be junk, when mutations develop in those areas, it can lead to serious problems. Say in the human genome, it can lead to cancer, diabetes, heart disease, various ailments like that.
We've talked a lot.
Okay, so there's actually a lot going on there in just that little bit. So he says the entire genome is appearing to be functional. That's not true. Like, again, he's just misrepresenting the findings. And if you read, again, the 2014 ENCODE paper that creationists ignore, you'll see that even they're not saying the entire genome is functional. They're very clear about saying that some of these regions that we're documenting biochemical activity are very likely to be non-functional, right? That's what ENCODE says. Now, the other thing here is that he talks about this like as a problem for evolution. And again, he's not being clear with what he's alluding to here, but he's hitting like all these very typical creationist arguments. The thing that he's alluding to when he says this is a problem, people are realizing this is a problem for evolutionary theory, is the Dan Grauer, the thing that Dr. Dan Grauer is very critical of ENCODE. He said very famously that if ENCODE is right, evolution is wrong. Now, what he meant by that was that, like, if ENCODE is right, then some of how we think about like genetics, we have to rethink that. But what creationists have taken that line out of context to mean is if ENCODE is right, then evolutionary theory writ large is wrong. So that's a misrepresentation of what Grauer said. But there's another level to that, and I've done a video on this, and I'll link that down below afterwards as well, because I just didn't think to link it beforehand, is that Grauer actually doesn't think that anymore. He published this work, so that was actually from a conference in 2013. He published this work in 2017 talking about the functional fraction of the human genome and how there would be an upper limit to that. But then another team took his work and actually found an error in it, published a correction in 2020. Grauer agreed with the correction, and if you read the 2017 paper, there's actually a correction appended to that paper. So not only is this creationist argument a misrepresentation of what Grauer said, Grauer, even if that wasn't the case, what Grauer said doesn't apply anymore because he has been corrected and he agrees with the correction. So I'll link that the video I did on that down below in case you want more details on that. Let's see, 15 minutes. Okay, so now we're going to just jump just 20 seconds here. So this is not necessary. Okay, so now let's talk about the last thing.
That point to our creator.
So this whole idea of the genome being a system of systems debunks evolution because it all had to be put together all at once for it to work, just like a computer has to be put together all at once for it to work, or an automobile or a washing machine. The genome had to be put together for it to function all at once, which included all these different categories of DNA that we've talked about. Now, evolution would say that the genome would have evolved bit by bit over long periods of time. But that just doesn't work because we know that it is irreducibly complex beyond our wildest imagination.
Wow. Well, thank you so much, Dr.
Okay, so that's where we're going to end it. I just want to comment on that last bit because he's just going straight to irreducible complexity here, which I've, again, done a video on this. I'll link it down below. It's so bad because irreducible complexity, the way he's describing it here is he's assuming that the current state of things, right, the current system must be the way it always existed, which, of course, that's not the case. The first replicators weren't representative of the replicators that exist now. The earliest cells would not be as complex as the cells that exist now. Everyone knows that. So he's saying the things that exist in their current state must have had to just poof into existence because of irreducible complexity. But we know that the earliest systems were not as complex as what we have. We've actually documented what those less complex systems would look like. We can actually find some of them in nature. You could look at any number of systems where it was originally claimed to be irreducibly complex, like an immune system or a blood clotting system or whatever. And you actually can find simpler versions, reducible versions of those systems that are still perfectly functional. He's also ignoring, by saying it has to work the same way as its current form, he's ignoring processes like acceptation, where something does one function and then it changes function and it evolves to do a different thing. He's also ignoring changing fitness landscapes, where something is beneficial in one environment but harmful in another environment. And as environments change, the evolutionary dynamics change. He's ignoring all these factors that facilitate the evolution of so-called irreducible systems. And I've done a lot of stuff on irreducible complexity. Again, I'll link it below. Man, that was terrible, wasn't it? That was really bad, but we're not done. Because the thing with ICR is really interesting. ICR, the Institute for Creation Research, is they really make a thing out of being like, we're the sciencey young Earth group. They don't tend to get involved in like culture war stuff. Like Answers in Genesis, AIG, they're just all about culture. You go to AIG's website or listen to Ken Ham, it's mostly culture war stuff. They went like all in on anti-vax stuff during COVID. Like they're out there. CMI, Creation Ministries International, they get involved with it, sometimes not in the way you'd think. Like they were actually vocally pro-vaccine. And that actually got them in trouble with their conspiracy theorist, young Earth creationist, anti-science audience, much to their dismay, which, like, what did you expect to happen, y'all? But ICR doesn't get involved in any of that. They're just like, no, we want to talk about it. We're just sticking to the science. So considering that they're just really all about the science, it's kind of funny that when I commented on this video, and I'm going to make this bigger so you all can read it. Let's do it this way. I commented on this video because, like, let's make this a bit bigger so everybody can see it. Just like pointing out, like, hey, you've got some problems here, guys. So I just said the human genome is transcribed from end to end. That's false. All pseudogenes regulate other genes. That's false. There are no neutral sites in the human genome. That's false. And like what I want to emphasize to their audience is anyone watching this should know that you're just straight up being lied to. Right. And I commented elsewhere on this. So I commented, Doki Doki Bible Club said something, and I because they referenced me. So I actually commented on there as well. So this is what I see. This is a screenshot of what I see on my YouTube when I'm logged in. Now, this is what you see if I open up like an incognito browser window where I'm not logged in. This is what I see. That's weird. I'm not blocked because there's my other comment, my innocuous comment, but my comment where I pointed out like some very simple, straightforward, and not controversial factual errors, that comment seems to have disappeared. That seems weird for the organization that seems to want to be all about the science and presents themselves as being specifically all about the science. They're all about the science as long as it's the science that they agree with. As soon as you point out very clear-cut factual errors, we don't want to hear that science. We're just going to delete that comment because we don't want to hear it. So that is what I have for you all this evening. I wanted to quickly go through that video. I am working actually currently on a longer junk DNA video. That's going to be my next creation myth, is going to be there's no junk DNA, and I'm going to give it a fairly comprehensive treatment. So if you're watching this in the future, and I have already published that video, it'll be linked down below. If you look down below and it's not linked, it means I haven't published it yet, but I will in the relatively near future. So before I go, I just want to thank a couple people. Thank you for the video, Dr. Dan, Star Runner. I appreciate you being here and your generosity. And Andrew Kroll, thank you. I appreciate you being here. So that is the show, everybody. This is this absolutely terrible, just wrong, dishonest video on junk DNA from Dr. Jeff Tompkins and Institute for Creation Research. I hope you found that informative. That's all I got. I'll be back soon with some more creationist debunking. Have a great night, everybody. Stay safe, and I will see you next time. Oh, before I go, please hit like and subscribe and like, comment, and share. Do all the things. Have a good night, everybody. Good night.